In this presentation, I would like to discuss single molecule biosensing with whispery gallery mode resonators. Life operates at the single molecule level, but most tools like conventional mass spectrometers and optical microscopes measure millions of molecules at once, thus averaging over the ensemble. There have been breakthroughs in single molecule biology, and for example, the Nobel Prize in Chemistry in 2014 in super resolution microscopy takes advantage of the single molecules to break the diffraction limit in conventional microscopes. In this slide, I'd like to share a few examples of why single molecule biology is important. One of them is protein folding dynamics. So proteins fold through multiple pathways, but we typically observe an average over these dynamics. So observing these processes at the single molecule level would lead into insights about how proteins could misfold, for instance. Similarly, a single viral particle can bind and trigger infections and diseases, and genetically identifiable cells make different fate decisions. In this landmark experiment showing stochastic gene expression, biologists expressed both red and green proteins in the same manner in E. coli cells with identical promoters. If both of these genes were expressed at the same level, then you would expect the cells to be yellow. And these traces are actually showing any external dynamics which cannot be controlled for. However, they found that the cells were colored from a range from green to red. And that is because inside the cell, intracellular, there is some stochasticity at the single molecular level, which flips a genetic switch, which is irreversible. The reason why I'm interested in about whispery gallery mode resonators is their application in single cell proteomics. So proteins in a typical mammalian cell, in fact, in this plot here, they're plotting the number of protein types and the number of copies for each of those proteins in a typical mice cell. And these proteins have a huge range of mass and a huge range of concentration. And conventional mass spectrometry has a detection limit at about 1 million copies of a protein. Since we can't amplify proteins the same manner that we can amplify DNA, any mass spectrometer requires 1 million copies of that same protein molecule to be sensitive. And so all of these proteins, which are known as the dark proteome, have to be inferred from mRNA levels and are not explicitly observed intracellularly. And so if we want to move forward for single-cell proteomics, we need label-free detection at the single molecule level, so single protein sensitivity, which is currently out of reach of current mass spectrometers. So single molecule label-free detection has been explored in many different techniques. And so here I'll talk about a few. One of the most powerful is surface-enhanced Raman spectroscopy, scattering. And here we're measuring molecular vibrational modes. So before we go on to these other techniques, I'll take a small deviation to explain SIRS. So first of all, Raman scattering is sort of inelastic scattering that happens in molecules. So in a typical molecule, you would have these energy levels, which S0 corresponds to the ground state and S1 corresponds to the singlet state. If you have a photon absorbed from this molecule, you would be excited, an electron would be excited from the ground state to the first excited state, which can then vibrationally relax and then fluoresce. However, you can also absorb photons in what are called virtual energy levels, and you have some sort of Stokes and anti-Stokes scattering, and this is known as Raman scattering. You can take the spectrum of absorption, fluorescence, and Raman to use as a footprint of that molecule. One thing to note, though, is that Raman scattering has very, very poor quantum yield. Quantum yield is the fraction of output photons to absorb photons. So while about 7 out of every 10 photons get fluoresced from a molecule once they are absorbed, Raman scattering is very, very inefficient. And so you need about from 1 million to 1 billion molecules to be absorbed from that molecule to then scatter via Raman scattering. So this is an example of a Raman spectra shift for water molecules. So you're typically measuring the shift from the excited wavelength, the absorbed photon. And these bumps correspond to these virtual energy levels. And for example, for water, these correspond to these vibrational modes, bending and symmetric stretching. What's very powerful about Raman spectra is that it's label-free and it creates a footprint of that molecule, which can be used to classify and ID that molecule. So as I mentioned, the quantum yield of Raman spectroscopy is very, very poor. However, there has been advancements via surface-enhanced Raman scattering. And what they're using here is they put samples on gold and silver metal spheres, which turn out to enhance the electric field after they are excited by a pump or absorbed photon. So here you have this near-field electric field enhancement via these gold and silver nanoparticles. So once again, we're enhancing both the incident and scattered photons, which increase our yield, as I had mentioned. And so it turns out that you can take a single molecule source spectra of individual molecules. And here in this paper, they showcase the spectra of 10 individual amino acids, showing the power of this technique. Another type of label-free detection is fluorescence correlation spectroscopy, where in essence you're measuring the diffusion coefficient of single molecules by measuring their residence time in a small laser focal spot. So as a molecule enters this focal spot, depending on its diffusivity, which is dependent on its size and mass, it stays more or less in this focal spot. By autocorrelating the traces of these single molecules, you can get a measure of the diffusivity, which gives you a measure of the mass and size of these single molecules. You also have nanopores, which can be used, for example, in DNA sequencing, where now you're passing molecules through a very small pore and measuring currents across this membrane. These ionic currents can give you a signature of the molecule, which you can use to detect and ID single molecules. And finally, what I'm excited about is whispering gallery mode resonators. And here, we're essentially measuring the refractive index of single molecules. And I'll discuss how that's done in the rest of this presentation. So resonators for biosensing are not a novel idea, but they're extremely powerful. And the reason is because a lot of materials and anything really has resonant frequencies, and this resonant frequency can be very sensitive to changes in their environment. So for example, in this paper, in a very clever technique, they take a steel tube with about 1 millimeter wide and a few 10 centimeters long, and they're passing cells in a fluid inside the steel tube. And this steel tube is essentially measuring the mass inside at a single point. So these particles are sparsely distributed, and if you can measure the resonant frequency of the steel tube, you can get a signature of the particles inside the tube, which are a lot smaller than the tube itself. The way this works is by actuating one side of the tube via piezoelectrics and sensing the frequency response on the other end. In this experiment, they're measuring the water content of tumor spheroids, which is a good biomarker of drug response in cancer treatment. Another idea from Stanford of Amir Savi-Laini's lab is using nanomechanical mass resonators for exactly single-cell proteomics. So now you have a chip with many, many nano resonators, which you can measure the frequency of, and as single particles bind on the surface of these resonators, you can measure the mass change of this resonator. And they very cleverly found out that they can do this by multiplexing many, many mass sensors, thus being able to measure millions of proteins per second at the single Dalton sensitivity, which is 10 to the minus 24 grams. So very impressive work using resonators for single molecule biosensing. So whispering gallery mode resonators use whispering gallery waves, and this were first described by Lord Rayleigh in the 1800s, where in St. Paul Cathedral's London, he was able to hear a whisperer on the other side of the cathedral. Because the waves, the acoustic waves, are totally internally reflected across the cathedral and thus concentrated to the listener on the other side. So total internal reflection you might have heard of before, but in essence, uh, below a critical angle, light gets both reflected and transmitted or refracted. Above a critical angle, this is totally reflected. However, there are, because of the boundary conditions of Maxwell's equations, there is a resident evanescent wave at the surface of this material. And so here I derived this evanescent wave, and what is happening above the critical angle at which light is totally internally reflected, you have this evanescent wave, which is propagating along x, yet exponentially decaying along y with no oscillation. And so this means that you can actually couple light from outside of the surface via interaction with this evanescent wave. And this is what whispering gallery mode resonators take advantage of. As another side brief, I'd like to talk a little bit about optical cavities and how they work. So optical cavities resonate based on constructive interference inside the cavities. So for linear cavities, this is the equation, where in essence you get constructive interference when integer modes of that wave add up. In ring cavities, you have the circumference as your optical path length. If you can measure the transmission or spectrum of these cavities, you find that at very specific frequencies, they're constructively interfering, and thus you get very tightly peaked resonant frequencies as shown here. And because they are of integer wavelength, you get evenly spaced resonant frequencies if you can measure the transmission spectroscopy across these cavities. The distance between these peaks is known as the free spectral range, so this distance here. And the quality factor is a dimensionless parameter which you can use to assign a certain cavity, which describes the losses of that certain cavity. And this describes the ratio of the resonant frequency over the line width. So higher quality factor cavities have less losses and vibrate longer. In whispering gallery mode resonators, you can excite the cavity by bringing the small bead close to a tapered fiber, and this tapered fiber will couple via these evanescent waves to the optical cavity. And so by measuring the spectrum, you can measure the resonant frequencies of your cavity. So these whispering gallery modes can be made of various geometries, and they have extremely high quality factors. So this really amplifies any sort of light matter interactions between the cavity and anything outside of the cavity. Because of their extremely high quality factor and low loss, the photon lifetime inside these small cavities of maybe 200 micron size is 10 microseconds, or essentially a same photon can propagate for 200 meters inside a sub-millimeter cavity. This is really the advantage of using whispering gallery mode resonators, because you have amplification of any sort of light matter interactions because of this long residence time. And if you can measure the transmission spectra of your cavity, you find very strong sensitivity to single molecules binding to the surface via different methods, which I'll explain in the next slide. But first, let me talk about the experimental setup. So the way you would set up these whispering gallery mode resonators is you need to first find a source with which you can sweep a wavelength, thus creating the spectrum. The way this can be done is through diode feedback lasers, where now you're changing the wavelength of these lasers by sweeping in current. Since as you increase more current, there is an increase in heat and temperature, and you can actually change the refractive index inside these lasers, known as the thermal optic effect. And it turns out that you can relate current to very small shifts in wavelength. So this is the trick that experimental users use to sweep wavelength. So in essence, you get a wave function generator, and you make a sawtooth current profile and feed it to your laser. Then you couple this light via the glass bead, as I mentioned, with these evanescent waves. The glass bead you can make out of standard silicon oxide glass. And as sample molecules bind to the glass bead, you have a shift in your transmission spectra. The way you measure transmission spectra is by taking a photodiode and simply measuring the intensity across this fiber optic as a function of the current or wavelength. Now, of course, the varying current also varies the intensity of the laser, and so it would look something like this. So you can normalize it with intensity, and you get your transmission spectroscopy with these peaks of resonance. The resonant frequency peaks are very sensitive to their external conditions, and exactly this change or this resonant shift which you can measure experimentally. So here we have single molecules binding to a gold nanorod on the surface of a whispering gallery mode resonator. And as this molecule binds to the surface, you get a small shift in your resonant frequency, which you can measure. Similarly, because there are whispering gallery mode resonators that support both clockwise and counterclockwise waves, a particle or molecule landing on the surface of this whispering gallery mode can produce some sort of asymmetry, which causes the mode to be split in a process known as mode splitting. And once again, measuring the delta between these two different resonant frequencies can give you an indication of how much material is bound to your resonator. So now going into some practical experimental applications of WGMs, you can use WGMs to detect single virons. So this is really outstanding because these whispering gallery mode resonators are although they are quite big, they are about sub-millimeter, you can detect single virons which are 90 nanometer scale. And here they show that by measuring this resonant frequency, you can measure discrete steps of viral particles binding and unbinding on your whispering gallery mode. So here it's an SEM image of these single virus particles landing on the glass microsphere. Similarly, you can measure in real time enzyme conformational changes. So now the enzymes are bound to a golden nanorod bound to the surface of your WGM. And as the enzyme changes shape, you can measure this change in shape by measuring the resonant frequency of your microsphere. And so these asterisks show discrete steps in this enzyme conformational changes. Now you can actually use, in this paper, both surface-enhanced Raman scattering and whispering gallery mode resonators to amplify and enable a higher sensitivity of Raman spectroscopy of single molecules. So in typical Raman spectroscopy, you would excite via pump and measure your scattering. In SIRS, you actually have this local near-field enhancement due to surface plasmon resonance of gold and silver materials. Now in WGMs, you can further amplify your pump because it is being circulated in resonance of your microsphere. And so you get, in this paper they showed they can retrieve 30x enhancement of just SIRS spectrum using WGMs in combination with SIRS. And so these are Raman spectra of single molecule. What's cool about this technique is that you can do 2D imaging because you can shift both your sphere and your signal collection across a glass slide with many different structures to amplify or bind single molecules of interest. So you can actually make these spheres in the lab quite simply by using a pipette puller. So this takes a glass rod about 1 millimeter wide and 10 centimeters long and pulls it at a very slow rate while heating the center of the rod. This causes the rod to taper until it breaks and you get two perfectly tapered glass fibers. And then you can use a similar instrument which heats the end of your tapered fiber. And because of surface tension, the end of your tapered fiber actually makes a perfect glass sphere, which can be used as your whispering gallery mode resonator. So here I actually show that I can couple laser light into this microsphere. However, I don't have currently the right laser and photodiode to measure the transmission spectroscopy of my whispering gallery mode resonator. Here are my references. I hope you learned something from this presentation. I'm happy to answer any questions in the comments. Thank you for listening.